Hey everyone, welcome back. I'm really glad you're here. Today we're going to explore DNA as the programming language of life. By the end of this lesson, you'll never look at code the same way again. Because what you're about to see is that nature has been writing software for billions of years, and it's actually pretty elegant. Let's dive in. Before we begin, here's what you'll understand by the end of this lesson. You'll learn what DNA actually is, why it only uses 4 letters, how it stores information like a giant Git repository, how it packs itself into chromosomes, and why all of this matters for understanding m6A modifications later in this course. All right, let's start with the big question. What is DNA? DNA stands for deoxyribonucleic acid. But honestly, that name doesn't help much. Here's a better way to think about it. Imagine your entire body is a massive software project. DNA is the entire Git repository. Every gene is like one Python file. RNA is the program that gets loaded into memory when the cell wants to run something. And proteins are the final output, the results produced after running the code. Unlike the code on your laptop, DNA is a physical molecule. It lives inside the nucleus of almost every cell in your body. But the beautiful part is that it works a lot like software. Now here's something mind-blowing. A normal book uses 26 letters. Your computer uses just 2 symbols, 0 and 1. But life only needs 4 letters. Those letters are A, T, C, and G. That's it. Just 4 characters. With those 4 letters, nature has written the instructions for every living organism on Earth, from tiny bacteria to blue whales and even you. Think about that for a moment. Your entire genetic code is essentially one enormous text file written using only a 4-letter alphabet. Let me introduce those 4 letters properly. A stands for adenine, T stands for thymine, C stands for cytosine, and G stands for guanine. There's one important rule that makes everything work. A always pairs with T, and C always pairs with G. They fit together perfectly, almost like dance partners connected by hydrogen bonds. This pairing isn't random. It allows DNA to be copied accurately every single time a cell divides. So how is this code actually built? DNA is made from millions of tiny building blocks called nucleotides. Think of each nucleotide as a Lego piece. Each one has 3 parts. A phosphate group, which forms the backbone. A sugar molecule called deoxyribose. And one of the 4 bases, A, T, C, or G. Millions of these Lego pieces connect together to create one incredibly long DNA strand. But DNA doesn't exist as just one strand. It has a partner. Together they twist around each other to form the famous double helix, which looks like a spiral staircase. The bases stay on the inside, connecting the 2 strands together. The sugar phosphate backbone forms the outside, like the handrails of the staircase. This structure is extremely stable. And that's important because your DNA has to survive for decades while protecting your genetic information. Let's see how base pairing works. Imagine the top DNA strand contains the sequence ATCGGTAC. Using the pairing rules, the opposite strand must be TAGCCATG. Every single letter has exactly one matching partner. No exceptions. This simple rule allows your cells to make nearly perfect copies of DNA every time they divide. Now you might wonder, why does DNA need 2 strands? Wouldn't one be enough? Here's the clever part. Having 2 strands means one acts as a backup. If one strand becomes damaged, and this happens all the time because of sunlight, chemicals, or normal wear and tear, the cell simply looks at the other strand to figure out what the correct sequence should be. It's like having built-in error correction. Nature solved data integrity problems billions of years before computers existed. Now here's another amazing fact. Every single one of your cells contains about 2 meters of DNA. But the nucleus, where all of that DNA is stored, is only about 5 to 10 micrometers across. So how does 2 meters of DNA fit into something so tiny? The answer is careful packaging. DNA wraps around proteins called histones, almost like thread wrapped around a spool. Those spools fold into even tighter structures until they eventually become chromosomes. It's one of the most impressive data compression systems in nature. Let's zoom out and see the entire hierarchy. You have a cell. Inside the cell is the nucleus. Inside the nucleus are 46 chromosomes arranged as 23 pairs. Each chromosome is made of tightly packed DNA, and scattered along that DNA are genes. It's very similar to the way software projects are organized into folders, files, and functions. So what exactly is a gene? A gene is simply a section of DNA that contains instructions for making something useful, usually a protein. Using our software analogy again, your entire genome is the repository. Each gene is like an individual source code file. One might represent login functionality. Another might handle payments. Another might manage user profiles. The important thing is that cells don't use the entire genome all at once. They only activate the genes they need at the right time. A skin cell and a brain cell contain exactly the same DNA, but they use different sets of genes. That difference is what makes them completely different cell types. Now let's look at some numbers. The human genome contains roughly 3.2 billion base pairs, and it contains around 20,000 protein-coding genes. But here's something surprising. Only about 1 to 2% of your DNA actually codes for proteins. The remaining DNA controls when genes turn on, where they turn on, how strongly they turn on, or serves structural and regulatory functions that scientists are still discovering. As computer scientists, this next idea should feel very familiar. Computers store information as binary. DNA stores information as combinations of A, T, C, and G. Both are digital information. They simply use different alphabets. This is exactly why bioinformatics exists. Sequencing machines convert physical DNA into long text files containing nothing but A, T, C, and G. Then computer scientists use algorithms, statistics, and programming to analyze that information. Here's what a sequencing read actually looks like. A red identifier, a sequence of DNA letters, a separator, and quality scores that tell us how confident the machine is about each letter. Everything we do later, finding mutations, measuring gene expression, or detecting m6A modifications, starts with these simple strings of A, C, G, and T. Now I want to introduce an idea that connects today's lesson to the rest of this course. So far we've been talking about DNA, but biology becomes even more fascinating when we move to RNA. Inside RNA, one of the bases, adenine, can receive a small chemical tag called m6A or N6, methyladenosine. This does not change the DNA itself. Instead, it changes the RNA message. That small chemical modification can influence how long an RNA molecule survives, whether it gets translated into protein, and where it travels inside the cell. In other words, cells can fine-tune their software without changing the original source code stored in DNA. We'll explore this idea much more deeply in the coming lessons. Before we finish, let's quickly summarize everything we learned today. DNA is the long-term storage system for life. It uses only 4 letters: A, T, C, and G. Those letters follow strict pairing rules. DNA forms a stable double helix. It is tightly packed into chromosomes. Genes are functional sections of DNA that contain instructions. And sequencing technology converts this biological information into digital data that computers can analyze. Most importantly, modifications like m6A show us that biology doesn't just store code. It also edits, regulates, and fine-tunes the messages produced from that code. Now let's test your understanding. Can you answer these questions? What are the 4 DNA bases? What 3 parts make up a nucleotide? Why does DNA have 2 strands? What is the difference between a gene and a chromosome? How many base pairs are there in the human genome? Why do we compare DNA to computer code? And finally, is m6A a DNA modification or an RNA modification? Pause the video for a moment and see how many you can answer. Here are the answers. The 4 DNA bases are A, T, C, and G. A nucleotide contains a phosphate group, a sugar, and a nitrogenous base. DNA has 2 strands to provide accurate copying and built-in repair. A gene is a functional section of DNA, while a chromosome is a tightly packaged DNA molecule containing many genes. The human genome contains about 3.2 billion base pairs. DNA is compared to computer code because both store digital information using symbols. And finally, m6A is an RNA modification. If you got most of those correct, great job. In the next lesson, we'll answer one of the most fascinating questions in biology. If every cell in your body contains exactly the same DNA, how does one become a skin cell while another becomes a brain cell or a liver cell? We'll explore gene expression and discover how cells decide which parts of the genetic code to run. That concept is essential for understanding everything that follows, including m6A biology. Thank you so much for watching. If you enjoyed